Okay, first I'd like to thank the organizer for giving me the opportunity to present my current work here. So my name is Yao. I'm from Dr. Califano's lab. And the title of my presentation is Compound Functional Mode of Action Identifying Compounds that Affect Transcription Factor and Signaling Molecules Activity. So in fact, this is a specific example and application of the virtual proteomics method that Mariano has just talked about. So he has gave us a great and comprehensive background, and I won't talk too much about that now. So the aim of this story is to first to predict the drug mode of action based on gene expression using the reverse engineering computational tools in our lab, and also the virtual proteomics method. And the second aim is to predict the new target drug interactions and also validate that through the experiment. So the general approach is first we construct the arachidine network using the gene expression profiles of a certain cellular context, for example, some cell lines or a certain tissue type. And then we use the signatures. So for this study, it is the compound perturbation data. And we then use the virtual proteomics to infer which transcription factor or drug target are modulated by this compound so that we can infer the drug mode of action. So in order to validate this approach, we use either the existing known drug target information from some database, we predict new interactions and validate that through the reporter gene assay. So the first step is to construct the network. So since the drug perturbation data is from CMAP MCF7, we want to construct this specific network. So we use the expression profile from CMAP MCF7 drug perturbations, but however, the variation in the expression for this data set is not large. As a result, the infer network is not large. We cannot infer regulance only for a limited number of transcription factors and drug targets we are interested in. So as a result of that, we also construct a network for the TCGA RSG breast cancer sample, and we integrate both of them. So in this sense, we both cover a large space of the target we are interested in, and also we keep the specificity in this network. So it includes around 1,800 transcription factors we currently know, and also around 2,000 drug targets in the drug bank. So for the signature, we use the CMAP MCF7 cell line. It contains about more than 3,000 samples for 1,293 unique drugs, which means that there are replicates for quite a few of them. And this is good news, since only based on these replicates, we can know that whether those drug perturbation data are reliable or not. So we want to first filter out the drugs whose replicates are not similar to each other. So we do this based on measuring their distance using the gene expression analysis, using the top upregulated genes and the top downregulated genes. So this is the distribution of the distance between all sample pairs. So as we can see, only a few number of the sample pairs are either highly similar to each other or the opposite to each other, which means they have some related mode of action. And then we calculate the percentile of the drug replicate distance in all sample distances. So a total of 1,163 drugs out of the 1,293 drugs have replicates. As we have expected, most of the replicates are similar to each other. So for example, this one, cyproterone, the percentile of the replicate average distance is 0.016, which means they are really similar to each other. But however, there are a few drug replicates that are not similar to each other. So this might be because they have different concentrations in the experiment, so they have a different expression profile, or just because there are experimental noise. So we should exclude those ones that are not reliable. We set a cutoff around 0.4 here. So we just analyze the drug signature that is better than this cutoff. So we integrate those the replicates. As a result, we have a unique signature for a certain drug treatment. And for the drug target information, we use the drug bank, which is a highly curated database and contain comprehensive information. the overlap between the CMAP MCF7 with the drug bank is 178 drugs with 103 targets. So those drugs are already filtered. Then we ask whether we can filter out some target that is not expressed in MCF7 at all. So we use the MCF7 RNA-seq data in a GEO dataset, and we just keep the targets that have a raw count number larger than 20 to make sure that those targets are expressed in MCF7. And this leaves us 120 drugs with 87 targets. So I want to emphasize that this selection of this dataset is unbiased process. And then we have the specific network. We have the drug signature. And then we use this virtual proteomics to infer the master regulator activities of each of these drugs. So now we have two profiles. One is the gene expression profile, and the other one is the master regulator activity profile. So we want to ask questions. First, can we cluster drugs with the same target that have the same mode of action, either using the gene expression or using the master regulator activity from the virtual proteomics data? So as we see here, both of them are good in clustering the drugs. That the drugs that have the same target, they are clustered together. So the second question we want to ask is whether we can infer drug mode of action based on gene expression. So to do this, we analyze for each target. We have a number of drugs known from drug bank that that modulate this target. We want to see whether this drug will induce larger changes in the mRNA level compared to the other drugs. So we rank those drugs. So for each row here, it is the target. For each line, it is the ranking of the drug for this target. So what we expect is that they should be enriched in the top rank, but this is not what we see. So after the integration of all the targets, there's no significance at all. So we think that the mRNA level is not, does not correspond to drug activities, and we cannot infer drug mode of action directly based on mRNA levels. So then we asked us whether the master regulator activity we infer from virtual proteomics can infer drug mode of action. So instead of checking for mRNA levels, we check the master regulator activities from virtual proteomics. And this is what we get from here. So as we see, for most of the target, it has good enrichment for the real drugs for those targets. And the key values for all the integrated result is highly significant. So we think that this master regulator activity of the drug target we compute from virtual proteomics is a good way to infer the drug mode of action. So although most of them gave us good results, there are some of them that didn't work out. So some reason might be the concentration of that drug didn't reach biological activities like IC50, so it doesn't have a really effective signature. Or because like for HDAC inhibitors, we don't know the specific subtype for them, so this is not really correct. So I just show here the same way that we did for the target with raw counts fewer than 20. So as you see, there's no significance at all since those targets are not expressed. And this is a specific example for ESR1, as Mariano mentioned, that for the antagonist of estrogen receptors, we did see significant differential activity for ESR1, and the higher the concentration, the higher the differential activity it is. However, for the agonist, we didn't see a good result here. So the reason probably is because that this estradiol is already in the culture media of MCF7. So this pathway is already activated. Even though we add more agonist, it doesn't make much difference there. So this is the reason why we mainly focused on the antagonist of the drug target. So now we have proved that we can predict drug mode of action and validate that through the known database. So the second approach is we want to make predictions of new drug target interactions especially for the transcription factor, since it's a very interesting protein family, as it is involved with lots of disease, cancer, and also cellular development process. But however, for traditional drug discovery, it's very hard to design small molecules for them, since the binding site for these transcription factors to the DNA, they are really large and shallow. However, using the virtual proteomics, we can predict new drugs that functionally affect the modulate transcription factors. So we did this, and we validate using the reporter gene assays. And I want to show the STAT3 example here. So these 4 other compounds that ranked top from the virtual proteomics that affects the activity of STAT3. And we use the reporter assay of STAT3 to measure the activity. So these are the 2 major figures you need to look at. This is the control, and the static and the glialactone are the known inhibitors for STAT3. And the green one is 2 hours, blue one is 6 hours, red one is 24 hours. So as you can see that our predicted compounds, it cause significant change in the luciferase signal of STAT3, and it's comparable, even better than the no inhibitors. So the conclusion of this story is that we constructed a cell-specific network for the breast cancer MCF7, and we successfully infer the drug mode of action using proteomics. And also we predict new relations between drugs and target, and we validate that for STAT3. So here, I want to thank Dr. Califano for his instructions and support, and also especially thank Dr. Mariano Javier since we worked together on this project, and he helped me with lots of problems, and also Eureka and Scale for performing the STAT3 experiment. And thanks for your attention.